Hi, good evening, everyone. Um, am I audible to each and every person?
Yes, ma'am.
Okay. So I am Upasna, and what Rohit has just mentioned that I am passionate about teaching and not much about research. So it's the true, but the research I include in the teaching, and generally I do the project-based teaching. And as Deeptanu has suggested to go for this commercial plant tissue culture, and tissue culture was always my favorite when I was doing this MSc and PhD research. So I am not giving any the technical details like related to my research work or anything. So it's just will be the informations like how commercial plant tissue culture works mainly in India, and which crops are more being getting in commercial plant tissue culture. So I will briefly give about two crops like sugarcane and banana and orchids, and then how industries are working. So let's start. So we'll move forward. This slide is not changing. Change here. Okay. The slide is changed.
Yes, ma'am. Yes, ma'am. It's
Okay.
changing.
So the flow of the lecture will be like first introduction what tissue culture is, and then the types of plant tissue culture and overall step how a successful plant tissue culture can happen, and then transforming agriculture through the orchids and banana and the secondary metabolite productions, and then success stories in India related to the farmers and as well as the industries, and then overall conclusion. So we'll go through first introduction. I think there are many agriculture graduates who might be knowing the exact plant tissue culture definition. It's like three, four words we have to remember if you have to write what a tissue culture is, like aseptic condition, explants, and the in vitro method and the sterilization. So those three, four words combiningly is a plant tissue culture. So why this tissue culture concept came? Like this is because the plant has the ability of totipotency, and what a amazing species is the plant, like it can do whatever it wants. So that morphogenesis, totipotency, differentiations, and regeneration capacity of plants getting used here for the tissue culture purpose. And when you will go through the timeline of like how plant tissue culture happens over the time period from 1900 to till 2020s. So father of tissue culture, as we everyone know that it's Haberland. So he has come up with the first concept of totipotency, and now which is rapidly used in this plant tissue culture. So gradually then the medium comes like how to grow the plants, and then in the medium also hormone plays a great role, like auxin cytokinin plays a major role in the plant tissue culture, like whether you have to go for callus, whether you have to go for the roots, whether you have to go for the shoots. So based on that also the hormones concentration also like it plays a major role, and as well as the compositions of the media, like plant cell we know they needs the major salts and the minor salts. So what should be suitable compositions of the media for different cultures. So then gradually it come up to the different cultures of the tissue culture, like ovule culture, ovary culture, anther culture. And so it was surprising for me, like till now I was remembering that the father of anther culture is Guha and Maheshwari, and those are the Indian botanists who have done this tissue anther culture work first. So for me, what surprising was like till now I was thinking, so Guha is a like male botanist, but uh, when day before yesterday when I was studying this history, I got to know that the this Guha, Dr. Guha is a female botanist. So it was quite surprising for me that like it was just in mind that we are thinking that the male are the like the men who are doing this tissue culture work or the scientist works. So next is the thing like come up with the like how we can incorporate the genetic engineering. So before setting up biotechnology or genetic engineering lab, first we have to set up is the tissue culture lab. How you will generate a transgenic if you don't have the tissue culture lab? So now the genetic engineering gets incorporated into tissue culture. So we are developing the genetic modified crops or genome edited crops. So now in the recent era, as genetic modifications is getting so much like ethical and these restrictions, so genome editing is getting the green signal. So now genome editing, the CRISPR-Cas9 technology is also getting included in this tissue culture to make more the desirable products, and as well as for the medicinal purpose as well, for productions of the secondary metabolites of plants which are having pharmaceutical properties. So this is a overview of the tissue culture, how it emerges and how it is now in the present situation. Next, we'll go for types of plant tissue culture. So, as we all know that the main thing in tissue culture is the explant. So, the tissue culture is the explant culture. So, what can be the explant? Based on that, there are the different types of plant tissue culture. It can be organ culture where you can use the root or shoot or a leaf, and it can be a seed culture where you can use the seed as your explant. It can be embryo culture where you can directly use your embryo as the explant. And next, this is a overall view of the plant, the different plant parts, how it can be used for the multiplication of shoots, and nodes also can be used for the meristem culture as well. And there are different process of getting an plant from the explant. So it can be through the direct morphogenesis or direct embryogenesis, or through the callus formations. So different processes are there when you are developing a plant through the regeneration process from the stock plant by using the tissue culture technique. And the seed culture and the embryo culture, mainly those cultures are used when there is the embryo abortions, and there is incompatibility and the embryo gets sterile, so there will be no regeneration, there will be no productions of the plant. So in those cases, to avoid those things, embryo abortions and sterility, we are using seed culture and as well as embryo culture. So next is the haploid productions. So as we all know that in marker assisted selection, double haploid population is used for this. So how these double haploid populations gets developed? So for that we need haploids. So how these haploids will come? And these haploids will come from these microgametes and the macrogametes. So what are those microspores and macrospores? So they are the pollens and the ovules of the plant. So through those, by doing the cultivations of these ovule and pollens through the tissue culture, we are developing the haploids. And the haploids also gets converted into the double haploids by using the colchicine. And in anther culture also, like in the when we are coming to the microspore, we are using either anther or the pollen. So there is a difference also using anther and pollen culture, because sometimes there will be inhibitors in the anther which did not allow for the pollens to germinate. So better to go for the pollen culture rather than anther culture. So pollen culture, it becomes more successful compared to the anther culture. So these cultures are used, and for them also specific medium is a nutrient medium is also used, and that also developed by the scientist. So next is callus culture. So what exactly callus culture is? Callus culture, everything is callus culture, because you have to at the if whatever you are doing using explant, you will come to the callus. And the callus phase is the necessary phase in a tissue culture technique. And this callus culture generally used for then either for the shooting or for the rooting, or then it will be used for the cell suspension culture, or it will be used for the generation of clonal variants. So what is somaclonal variant? So somaclonal variation is like variations mainly we are getting due to the crossing over, or due to the cross pollinations, or due to the some genetical changes. But that will be mainly because of the reproductive cells division. But when you are coming for the somatic cells, if you are getting any variation, so that is known as the somaclonal variation. So what is the reason behind these somaclonal variations? So prolonged culture, if you will do, so there will be the effect of the chemicals and there will be the environmental effect. and there will be the epigenetic effect. So epigenetic effect mainly is the histone methylation, histone modifications, so which causes the changes in the DNA structures and as well as the changes in the morphology, and that because of that the variations happens. So that is known as the somaclonal variations, and now these variations are getting generated intentionally to get the resistant varieties, and as well as the like this in sugarcane also the ono variety gets developed for the disease resistance. So this is a technique where this callus culture is used. And next, from the callus culture, uh, we will get to the cell suspension culture. So in which case this cell suspension culture is used, like when we have some chemical compounds which is necessary for our requirement, like whether it is having any pharmaceuticals or nutraceuticals advantages. So how to extract those chemicals from the plant cells? So better to go for the cell suspension culture, and we are selecting the best cell line out of the those callus, best cell callus, and we are going for the suspension culture that we are doing in the liquid medium, and to enhance the productions of those metabolites. Nowadays we are doing that in the bioreactors, so where we are multiplying the productions of these secondary metabolites, and then we are doing the downstream processing to get the exact metabolites in the sufficient amount. So in these cases the cell suspension culture is used, and but that for that also the callus culture is the first. So from the callus culture, we will go to the cell suspension culture. So next is protoplast culture. So what exactly protoplast is? So you know that in a cell without cell wall is the protoplast. What we will get without the cell wall. So how the cell wall will be degraded, and we know the cell wall is made up of cellulose and the pectins, and how we have to just degrade those cellulose and pectin to get the protoplast. So there are enzymatic chemical process and the mechanical process also, where the cell wall gets damaged and we get the protoplast. So mainly nowadays the chemical process is getting utilized, and that is you are using the cellulase and pectinase enzymes to degrade the cell wall. And now we got the protoplast after degrading the cell wall. So now we are doing this protoplast, isolated protoplast. We are plating these protoplast, and then the wall regeneration happen, then the division, then clump formation, then clone formation, and now it will go to the callus stage, and then there will be the callus differentiation. So now it depends on you, whether you want to go for the either shoot development, or whether you want to go for the fast root development. So generally what they do is, like, from first they go for the shooting medium, then they go for the rooting medium, and then they go for the plantlet formations. So this is the general steps in plant tissue culture. And the first, we know the mother plant plays a main role in the tissue culture, getting a good regenerated plant. So we have to choose the best donor plant from where we have to take the explant. Then after the explant gets inoculated, we have to do the then shooting, and then we will do the multiplications of the shoot. Then we will do take the shoot balls and the micro shoots to other cultures, like subculturing we will do, and then we will go for the rooting medium. And once the roots gets developed, then we have to now grow in the pot trays or in the this pots, so that you have to go in a controlled condition first to make it acclimatized. Then you have to go for the nursery production. So this is a like general overview for how tissue culture works, but it varies depending the conditions and the nutrient medium, as well as the timing, and how many times of culture should be done, which hormone should be used. Everything depends on the species to species, and as well as explant to explant. So then we'll come for the main this commercial thing, like how it happens in commercial scale, the tissue culture. So in India now, mainly they are focusing on the horticultural crops for productions through tissue culture. So first is orchid micropropagation, and we know orchid is a like everyone's favorite flower is orchid, and India has a diverse range of orchids, mainly in Himalayan regions. And how to grow these orchids was a very tedious job when they have thought of doing this tissue culture work, because the as the seeds germination happens when there will be the mycorrhizal connection on it, otherwise the seeds does not germinate. The seed has a symbiotic relationship with mycorrhiza, and then only after the fungal association, then only the seeds gets germinated. So that was bit difficult to find out like how to do this in vitro and aseptic culture of these orchid seeds. But later they come up with the ideas, and they did it by changing the medium and with the different not compared to MS medium, it was a completely changed medium which was used for this orchid micropropagation. And the main thing is, after the seeds gets germinated, then there will be the development of the protocorm like bodies, that is known as PLBs. So these PLBs are used mainly nowadays for as explant for orchid micropropagation. So there are different scientists who have tried different parts of the orchids for its cultivation through tissue culture. So some have used mature seeds, some have used protocorm, some directly protocorm like bodies. Before the protocorm gets developed, it will be like different undifferentiated mass, that is known as the protocorm like bodies. And then this is the overall procedure of this orchid micropropagation. So anyway, you have to first do this disinfection stage, the sterilization stage, whichever plant you are using. So you have to keep that on running tap water for the treatment with fungicides for 2 hours. Then you have to cleanse that with 70% ethanol. So these all are known as the surface sterilization. And sodium hypochlorite, we know everyone that sodium hypochlorite is a bleaching agent. So it is also used, and based on the concentration, the time duration varies. And then we have to wash with the sterile distilled water for 30 minutes to 2 hours. And then you have to go for the inoculation. And inoculation also varies depending on whether you are going for the liquid medium or semi solid or solid medium, or you want to have the more growth, so then you can keep that for the rapid growth in a rotary secker as well. And then culture condition, the culture condition is temperature is generally the room temperature 25 to 28 degrees Celsius, and light is 16 hours light and 8 hours dark. So these are the two commonly used basic medium for orchids, so Knudsen C medium and Vaccine and Vent medium. And the pH here used is the 4.8 to 5.0. So later I will show these compositions of this Knudsen C medium. So these are, see how many just minimum salts, but if you will go through the MS medium or half MS medium, so these are completely different. And there will be so many major salts and minor salt if you will see the MS medium. And the vaccine and went also, the little bit changes in case of the calcium sources and the iron sources. Otherwise it is more or less same. So so this is the medium composition what I have just now I have explained. And next is so now you have inoculated this orchid. So what you will do after the, you will get the shoot multiplications inside the orchids. So you have to transfer this from the flask to the petri plates, and then from petri plates you have to completely clean and remove the augurs which have been attached with these plates. So after the removal of the augurs, you have to transfer to this to the medium or the that is known as that cockpit. Those things can be used as, or perlites, those can be used as a growing medium instead of soil fast for growth of these orchids. And then there are different kinds of orchids also, like there are monopodial and sympodial orchids depending on their branches. So there are four ways like to propagate of these orchids. So these are different methods how we will propagate after that. So comes under the vegetative propagations of the orchids. And then you have to transfer to the soil. So there are different types. One is the hanging orchids and one is the soil based orchid. So you have to use the like based on the package of practice of the orchid, you have to use follow those protocols, and you have to use whether you have to go for the hanging or you have to go for the pot based growth of these orchids. And so this one is the hanging one, the previous one was the pot based one. And then there are also different maintenance of orchids, like you have when you have to water, the fertilizer applications and how to apply those fertilizers, then plant protection, which is a major pest in orchids and which is a major disease in orchids. So now it comes to the orchids pop, and based on the pop you have to maintain the orchid. And so this is the final overview of this. This is a final overview of the how banana cultivation happens. So this is a for the banana culture. So now just before we have completed the orchid, so now it is for the banana. So banana, the main problem is banana is, if you will culture banana pseudostem, you will get lot of compounds which will turn your medium into black color. So in banana multiplications, the main problem is the turning of browning of the medium. So how to remove those browning of the medium is a major problem here. So generally what they used to do is, while doing the sterilization, after sterilization they treat with antioxidants, because here because of the oxidation, the browning happens in the medium, and that makes the explant and the callus and the shoots everything brown. So before that itself they are doing this antioxidant treatments to make it like so that there will be little less of this browning of medium. And then after that then they will transfer and the meristem block to the medium, and there will be the initiation stage, multiplication, then root and shoot formation, and then acclimatization and growing in the polyhouse or greenhouse under control conditions. So while growing also there are different contaminations happen, so we have to remove that contamination. So better to do the prior treatment like the cleaning and sterilization. So while cleaning and sterilization itself, they use the bavistin and the fungicides to avoid the diseases. And at the end also, while doing this acclimatization stage also, they used to apply some like bio pesticides to prevent the diseases inside the polyhouse. So this is the overall flow of like how banana tissue culture happens, like removal and cleaning, then rinsing, the trimming. So trimming is like you have to take the rhizome tissue, and there is a exact size of rhizome gets used, and you have to sterilize with ethanol. You have to sterilize with sodium hypochlorite. Either you have to use sodium hypochlorite or you have to use mercury chloride. And but whichever time, how many you are using the sterilization, you have to do the washing with repeatedly with sterile distilled water. And then the rhizome will be ready, and then rhizome will cut around 1 to 2 centimeter, and that 1 to 2 centimeter uh, with the optimum side on the purpose. Okay, so the 1 to 2 centimeter will be fast, it will cut after all the sterilization process happens. And then each explant will be reduced to the size of 0.5 to 1 mm, because you have to make it thin slices to keep it on the medium, the growth medium. But when you will do the sterilization, after the sterilization you have to make it to the previous size what I have told you. And here the medium what is used is the basal MS medium, the general nutrient Murasik Cook medium what we are using for the tissue culture. And then this is the uh, sucker, and we are removing the layer. So if you have seen the banana rhizome, you might have seen there are different layers. So we have to go to the innermost layer before inoculating into the medium. So after that, the next process is when you have to remove this, and when you have to do the soft culture as well, and then the mass multiplications, and then the shooting, medium shooting, you will know that you need more cytokinin if you have to go for the shooting first, and then you have to go for the rooting by using the rooting hormones, and then agar weaning of plants. So like how in orchids we are removing the agar from the plants, here itself also you have to remove the agars from the explant or the plantlets, and then we have to transfer to that to the pot medium, into the polybags. So then here there are like primary hardening. First it comes primary hardening. So primary hardening is for the 4 weeks, and you have to keep the plant under 50% shade. And next is the secondary hardening. Secondary hardening is after the primary hardening, you have to keep another 5 weeks in the polybag. So after this, there will be the screening phase. So if you will see any variations, any morphological variations in the plants, then you have to discard that plant, because that is like a weed to your nurseries. And then the suitable plants will be packed, and it will be transported for the farmers or for the selling the seedlings. So this is the banana vegetative propagation, how the rhizomes are used for suckers are used for vegetative propagation. And this is the multiple shoots after the when the suckers are kept, the pseudostem is kept inside, then multiple shoots, and then shoot elongation, and the in vitro rooting, then weaning of agar, like after all the agars are removed from each plantlet, and then hardening, and then so this is the overall field view of tissue culture plants. So next come to secondary metabolite production. So what is a secondary metabolite? So plants generally produce secondary metabolites for its own benefits, like to protect from the stress, to protect from the pathogens, to get protected from the insects. So those secondary metabolites have pharmaceuticals and nutraceuticals values. So how to do those productions in large number? So there are medicinal plants and aromatic plants whose compounds are getting synthesized either in roots, either in stems, either in leaves or in flowers. So how to do the mass productions? In that case, now we are using this tissue culture medium. So in this, like earlier I just mentioned in the cell suspension culture, so here also we are doing the cell suspension culture to get our desired secondary metabolites. But the main process here is the downstream processing. So downstream processing is like to find out the exact compound what we are searching for. So that can be done only by the chemical screening by either LCMS or HPLC or GCMS or NMR spectroscopy. So then this thing helps us to get exact compound. And if you have to enhance the production, so you have to do the modifications to find out the target genes. We have to go for the sequencing. So after that we have to enhance that specific gene, and that specific genes after you incorporate, so that transgenic plant or the genetic modified or genome edited plant will be now grown in tissue culture to get the mass multiplications or mass productions of these secondary metabolites. And then those will get transferred to the bioreactors to or like rapidly enhance these productions. So this is a overall view of how secondary metabolite productions happens through this tissue culture, and it has a tremendous impact nowadays, because now many herbal industries which are having giving these herbal based products, they are doing this multiplication through this tissue culture. So now, and also another one thing is, there are some Ayurvedic organizations now, they are also targeting to do these secondary metabolite productions through the tissue culture, so that we will have much more number of plants, and we will save the plants which are getting endangered in the Himalayan range and as well as in the Western Ghat range. So this is an alternative solutions to get the plants like sustain, because there are some plants where the roots are the major sources of the secondary metabolites. So in that case, we have to uproot the plant. So that uprooting can be minimized if we will go through this tissue culture. So these are some success stories of the farmers who have adopted the tissue culture products in India and got awarded and got successful also. So these two are from the farmers who are using the tissue cultured banana crops and have been successful like till now. And this is one farmer who has developed a technique how to use leaves as explants, and he has got a patent also for that. So the farmers are also doing this work and getting the being becoming an inspirations for us. And there is like in Jammu Kashmir, there are the tissue culture labs now getting developed, and that's a new hope for the farmers over Jammu Kashmir. So how Indian scenario of this plant tissue culture. So mainly the how the industry works in plant tissue culture area in India. So first industry was established by 19 N.V. Thomas in 1987. It was in Kerala, and it was mainly for the cardamom productions. And after that, their major growth areas happened in Maharashtra, Andhra Pradesh, Karnataka, Tamil Nadu, and Kerala. So now I am in Bangalore. So I have seen many companies who are simply having a small space, but they are having large facilities of this tissue culture, and they are doing rapid success. So I have made the Thomas. I have given the photo, I think. So this one, this is the Thomas Biotech. So these Thomas Biotech, they are mainly working on the papaya productions. So he left his job and he started his own company, and now he's a big businessman in Bangalore who is doing this papaya cultivation. So there was a Bangalore biotech innovation was going on one 3-4 days conference. So there he got award as a like successful entrepreneur by doing this micro propagation of papaya. So now, if you know, this Tata Tea is also doing this tea tissue culture. Hindustan Liver is also doing Unicorn Biotech. So there are many more industries who all are sorry. So these are the list of industries, but these are the big industries, not the startups. So there are other startups also who are working in tissue culture, and now they are growing. They are in the growing phase. So now for this tissue culture also, government has started giving the support. So Ministry of Science and Technology, under the Ministry of Science and Technology, around 150 projects related to tissue culture for R&D and field demonstration is going on, and they have given so many supportive policies as well. So if over 50 commercial laboratories between 1987 and 1995, and total of capacity 210 million plants per annum. So these has happened because of the support from the Ministry of Science and Technology, and they are providing as well infrastructure and support facilities also, like for diagnosis of virus and the need for tissue culture, virus free and high quality plant material production. And so these are the bodies under government organization who are giving support for this tissue culture. So if you want to read in detail, like how much subsidies is given, how much monetary support is given, and how the, if you have a startup idea, how will get these grants. So this is the paper from where I have, sorry, from where I have taken all these informations related to the industry in India. So you can go through this paper, and if you want, I can share also this paper. So you will get a brief idea how the industry start, plant tissue culture industry gets started in India. And at the end, you have to go through this process, national certification system for tissue culture raised plants. Then only your product will be certified and you will able to sell your products. And this one I have explained you that how all these are the list of industries working on tissue culture in India. And this I have taken from ICR website. So now in Sikkim they have started the mass multiplication of orchids, and this is the by the Jain irrigation. So Jain irrigation earlier I was thinking it's for providing all the drip irrigation and sprinkler facilities, but now this Jain irrigation is also working on tissue culture and mass multiplications of the mainly horticultural crops. So now they have done with coffee farming by doing this tissue culture of coffee. And now in recently the in UP there was a banana tissue culture lab got established. So these all are the newspapers clips what I have attached here. And so this is a Kerala based company, that is Happy Biotech. So they have all the products which are developed by tissue culture. So mainly they are selling the orchids by doing the tissue culture work. This is a small startup in Kerala, and they are doing this work. And the more surprising thing for me today was like when I was going through this BioPrime. So it was in top 5 biotech companies in Bangalore. But what they are exactly doing, they are not doing any tissue culture products, but they have the tissue culture facilities. So by just providing the service also, they are earning profit. Like if some people they don't have facilities, they can go and do their work or some research work or project work over there. So this how a small idea is getting converted into startup. So it's quite amazing for me when I was reading and going through all these newses today. And next is there is the Terry is there in the Energy and Resource Institute. So they have a micro propagation technology park. So it's a new approach and a greener approach to provide support for the people who are willing to do the tissue culture work. So now let's come to this end. So it's like where I am working, we are having 5 departments. So we are mainly doing interdisciplinary projects. So I am working with mainly digital transformation and the interactive technology groups related to the predictive model and the predictive modeling, and as well as for the IoT based sensors, biosensors, all those things. And we have in life science department, we have a very good facilities for the instruments. So why I am sharing all this information is, so let's collaborate. So if anyone is interested to collaborate with me, because recently I have completed my PhD and I have not yet started any research. Just after my PhD I joined there. So I am willing to collaborate for starting any new research work, any industry related work, any startup ideas you have. I can also help in that. And so that's it. So together we can do more. So this is from my side for today. So end of presentation. Any questions?